Wow, cool exhibit.
Yeah, thanks.
I love science. Look at all these amazing exhibits.
Yeah, I always wanted to learn more about these things. Hey, look! Steroids!
Wow, those have been in the news recently because of all the sports scandals.
Let's go check it out. Hello, you like the exhibit?
Yeah, what was your category?
I didn't do creative writing, a model, or anything like that. I made my own category.
And what might that be?
Self-experiment.
Wow.
It is very good to see how it affects you. To accurately learn about steroids, I have been taking this human growth stuff since I was 7.
That's pretty cool. We're gonna go now.
Okay, have a good time at the expo. That was weird.
Yeah, it was really weird. I wouldn't want to do that for my biotech expo experiment.
Yeah. Hey, look! Someone brought an animal to the expo!
Hello there.
Hi,
What did you do for your category?
field trip.
Huh?
I take people on field trips to learn more about cloning.
Well, so where do you go on these field trips?
Inside eggs and cells. It's pretty sick.
Who are you?
I'm Dolly the sheep. I was the first animal cloned using SCNT. That's somatic cell nuclear transfer. It's a type of reproductive cloning.
Wow. So you don't have a dad?
Nope, I have a mom and a cell, but let me tell you a little secret. I don't like reproductive cloning.
You don't? But if it weren't for that, you wouldn't be alive.
True, but I really like this other kind of cloning. Actually, I have a secret passion for it. It's called gene cloning. It's much more interesting and fun than reproductive cloning.
That sounds way more scientific.
That's why it's so cool. Do you guys want to learn about it firsthand?
Sure!
Let's bounce.
Right now?
Yeah. And one other thing. In order for this to work, you'll need to assume the roles of different parts during the steps of gene cloning. I'll be your tour guide and follow you through this adventure.
Well, Sam! I'm a piece of DNA!
Same!
You must now be prepared. The restriction enzyme is coming to cut you up.
Hello! Ah! I'm here to cut you.
Don't be too afraid. When Mr. Restriction Enzyme cuts you, you'll be able to combine with another piece of DNA to form a recombinant DNA. Who knows? You two may even combine with yourselves.
Ha ha ha! I've now found your sequences. I can cut you. Now you both have sticky ends.
Yes, since your base pairs, David, are opposite of Sam's unpaired sequence, you'll stick together.
Wow, this is crazy! I'm a combination of 2 pieces of DNA!
Yes, you are, but your journey is far from over. You're now a recombinant DNA. You're a hybrid between 2 pieces of DNA, Sam and David. I will now call you Savid. Here, we'll just watch. Some other biotech kid came along earlier on this field trip, and now he's a bacterial plasmid.
Hello, I'm BP, short for bacterial plasmid. I'm a type of DNA just like you. No restriction enzymes have cut me.
You see, the restriction enzyme came back. It's making the small bacterial plasmid even smaller. To start out, bacterial plasmids are very, very small, usually only a few thousand base pairs.
I just gotta leave the same place I've been for a while. We bacterial plasmids have a single origin of replication. This means we all have to come from the same place in the molecule or we won't work. This means I have to wait around a while all the other bacterial plasmids are copying themselves.
Yes, he's right. The bacterial plasmid are very small and fragile subjects, and they'll not work unless they're all formed at the same spot.
Whoa! What are you doing?
I'm attracted to you!
Huh? I just combined with you!
Oh, I see what happened! Since we were cut with the same restriction enzyme, we can combine!
Because bacterial plasmid and SAVID are combined, we now call them BPS. It wouldn't have worked if you, bacterial plasmid, and SAVID were not cut with the same restriction enzyme. You wouldn't have the correct sequences.
But we did, so it worked out fine.
Yes, it does. Now the adventure of gene cloning gets a little more interesting. We're going to a party.
I love parties.
Yes, we're going to someone's home. The host's name is E. coli.
Guy or girl?
He's a guy, an old friend of mine. Well, let's get going.
Why isn't E. coli opening the door?
You see how you can't get in? That's because the host has his door completely shut. I will heat shock it for you. This is usually the scientist's job, but I'll do it today.
Yeah, we got in!
This is a nice place. Why, thank you.
Hey, E!
Hello, Dolly.
Long time no see.
Yes.
Anyway, BPS, this is E. coli. He is your host for tonight. What you just did is called transformation. You, the recombinant bacterial plasmid, entered the host cell E. coli. That is quite a feat.
You see, BPS, this is a national party night. All my other E. coli friends are having parties too.
You actually are going to stay here for a while. All these parties are happening on the same culture dish. So after you grow a colony up in the house, the gene cloning will almost be completed.
Yeah, while you're chilling down here, all the scientists up above will be scanning all of our parties and checking out all my guests. When they find the gene they want, they'll take you out of the party and examine you.
What if this happens to me?
Then you can't party. It definitely is a time to celebrate, though. You are finally a copy of a gene.
That's pretty cool. And I do love to party.
Me too.
Actually, we can't anyway. We have to go back to the Biotech Expo.
Whoa, that was crazy.
Yeah, it really was. Thanks for the adventure, Dolly.
Dolly?
Dolly. And the Biotech Grand Prize goes to Dolly the Sheep for cloning.